Hello students, welcome to EPG Pathshala. I am Devapriya Chaudhary from JNU. Today we are going to talk on the prediction of secondary structure of proteins under the paper Computational Biology. So when we talk about protein structures, there is a certain hierarchy in that. At the very basic level, you have the primary structure of proteins, which is nothing but the sequence of proteins. So then you have the secondary structure. This is the three-dimensional arrangements of amino acid residues in space, in which the structure is formed by amino acids which are close together in sequence space. So, for example, an alpha helix or an isolated beta strand, a beta turn, these are examples of secondary structure. At the next higher level, the protein chain, which now might be having secondary structures, then folds on itself, such that regions in the sequence, which may be very far apart in the sequence space, can come close together in the real three-dimensional space. So such a kind of folding results in what is known as the tertiary structure. Finally, many proteins are made up of more than one polypeptide, and when a particular protein has more than one polypeptide, these individual polypeptides are coming together and interacting in a very particular way, forming what is known as the quaternary structure of protein. So the next thing is that why do we have to predict? After all, it's quite possible to determine the structure of a protein experimentally. Common techniques used are X-ray crystallography, NMR spectroscopy, and nowadays even electron microscopy and electron diffraction. Now, if we can experimentally determine the structure of a particular protein, why do you bother to detect it? To predict it? The answer to that comes like this. So here this graph tells about how the number of experimental structures which are known till date are increasing with time. As you can see that the graph is kind of exponentially increasing. So every year on year, more and more new structures are coming up. Now that's a great thing. But if you look at the number of sequences in the next slide, you see that the rise in the number of sequences far outstrips the rise in the number of structures. So if 1,000 structures are, just for example, if 1,000 structures are being added every year, then maybe a million or many millions of sequences, protein sequences are being added to the database. Clearly, the number of structures which we need to determine must increase by many orders of magnitude if the number of experimentally determined structures have to come anywhere close to the number of sequences that we know. Now, given this deficit in information, the only practical way to fill it up, and which we need to do in most cases, is by prediction methods. And that's what this module is going to talk of. So, predictive methods, as I said just now, allows us to bridge this gap between the number of sequences and the number of structures. Even though predicted structures usually are wrong in certain details, but they're nevertheless quite useful in designing experiments. And if one uses them carefully, then structure prediction can be a very useful step in the design and sometimes interpretation of experiments, and that way one can minimize errors, we can minimize the time and the cost of doing research work. So different kinds of secondary structures, as I said. Here are examples of an alpha helix, a small turn, a beta strands. So these are the common examples. So the question is, how do we get to predict them? So before anything, let's give a formal definition of a secondary structure, and then we go onwards towards its prediction. So by definition, given a protein with residues, let us label them S1 to Sn, we need to assign each residue to a secondary structure class. And currently what one uses, the classes are for helix, which we call the H class, the strands, we call them the S class, And the coils, that is something which is not either a helix or a strand or a turn, we call them the C class. And finally the turn, which is again we call it the T class. Some programs give only a 3-state prediction, that is they omit the turn class, that is considered like coil. So they give you a helix, strand and coil, while some other programs they would give you a 4 class classification with a helix, strand, coil and turn. So what secondary structure prediction does not do, and that is also one has to be clear about. It does not produce an all atom model of the protein starting from the sequence. That's not its job. It gives no information about the relative arrangement of the secondary structures that it has predicted. Now let us say it predicts 2 helices. Now the helices might be parallel to each other or in some angle. It will not predict that. And no information is obtained on the individual strands which combine together to form a beta sheet. So if there are more than one beta sheets, we would not know which strand belongs to which beta sheet, or even within a beta sheet, the order in which the strands are coming up and pairing, we would not know. We would not be able to assign whether a beta sheet is parallel or anti-parallel. We will simply know that residues from this position to that position is a strand. Residue from this position to that position is a helix. Residue from this position to that position is a turn. And if there is no other prediction, then we'll assign it to coil. So now obviously if one has to predict something, there has to be some place where we have to match our results with. So of course this comes from the experimental structures. So there must be some way, by an objective way, by which given an experimental structure from X-ray or NMR or even EM, we can assign individual residues in the structure to a particular secondary structure. Now this is no means a very trivial problem. In fact, the definition of helices or strands have to be very precisely defined for a particular amino acid to be assigned to one or the other class. And there are programs like DSSP, which stands for the Dictionary of Secondary Structure of Proteins, another program called STRIDE. These are computer programs which have found immense popularity, and these, given an experimental structures, they will assign the individual residues to a helix, to a strand, to a turn, or to a coil, etc., etc. So if we just consider DSSP, it gives quite a detailed assignment. Say for example, it identifies a residue belonging to an alpha helix, to a 310 helix, to a pi helix, a beta strand, a residue belonging to a beta strand, belonging to a beta bridge, which is something like a single residue beta strand. For turns, it can be a beta turn, it can be a bend, which can be a different kind of turn, maybe a gamma turn or something, or it could be coil. Now most predictive programs do not provide such a detailed specification. Instead, all different kinds of helices are combined into one group like helix. The 2 different kinds of strands are combined into one thing, just a strand. Turns or bends are again combined into just simply turns, and coils, of course, which are essentially what are called the unassigned ones, they remain as such. So even though the assignment using DSSP, etc., would give you a large number of classes, prediction is usually restricted to 4 for some, and for some it's only 3, that is helix, strand and coil. The second important concept that we need to get clear about is the quality of the prediction. Now one could use an index called Q3 as a measure of quality, which is essentially the percentage of residues which have been correctly assigned in a 3-class prediction. So now since there are just 3 classes, if let's say I have a 100 amino acid protein and I randomly assign the amino acids to one or the other class, the chances are that I will be right is about for 33% of the residues. So a completely random prediction, Q3, is expected to be around 33%. So obviously any prediction method to be of any use must perform better than a Q3 of 33%. Now one should also see that 100% accuracy may not always be achieved, and not because of the fault of the prediction algorithm, but itself because of the fault of the method of assignment. Say for example, DSSP assigns a particular amino acid, usually would be at the end of a helix, as maybe as a helix, while stride might assign it to be as a coil or not as a helix. Now clearly that if we take different methods of assignment of secondary structures, there are some slight variations of the order of about 10% of the time when different programs do not agree, right? So therefore, when the assignment programs which are working with the absolute experimental data, if they cannot agree 10% of the time, one cannot blame the prediction programs if they also miss that. So about the maximum accuracy that what one can achieve by a prediction program is around 90%. Okay, that if we get about 90% accuracy, then we will consider it perfect prediction. So there are various different types of secondary structure prediction algorithms. The simplest one would be just a sequence alignment. Then you have statistical methods, which includes the Chou-Fasman and the Garnier-Osguthorpe-Robson method. More sophisticated methods are like the neural networks, which are there in the PhD server, or the Cypred and NPredict, and then again using hidden Markov model type of things like Cypred and SAMT98 and others. So there are various different algorithms using various different statistical mathematical computational framework, and they have the level of complexities are quite different, and the accuracy, as you will see at the end of these methods, are different. So given a particular use and the importance that we are assigning to secondary structure prediction, we might use one or the other program. So let's start from the simple end. So let's take the simple aligned model. Now essentially it is that you know a sequence, you know the structure, and you have it assigned. So let's say I have a protein, say hemoglobin, where I know that the first 16 amino acids form let's say an alpha helix. Now I take my unknown protein sequence whose secondary structure I want to know, and I simply do a sequence to sequence alignment between the one which I know and the one which I do not know, something like maybe using a Needleman-Wunsch type of algorithm. Now once I do that, now I see that that part of it which the first 16 amino acids and the part which aligns with it. Now if that one in the known structure is a helix, then clearly I would expect that the part which it aligns with in the unknown sequence also should be an helix. So essentially what we do, we align two sequences, one of which has known secondary structure assignment, and then we transfer the known assignments from the template to the aligned part in the target. Now this of course will work when the two proteins being compared are very highly similar. So 40 to 50% identity is at the minimum between the template and test sequences. Otherwise, as we had seen in earlier modules, that the accuracy of the alignment process itself is rather weak. So particularly when the identity falls below, let's say 25%. In such case, this method cannot be used for secondary structure prediction. Now let's come into the statistical methods, which just take a single sequence and proceeds from there. It does not need to compare with something like that. One of the earliest such method came to be known as the Chou-Fasman method, was proposed way back in 1974 and is nowadays considered more as a classical method. I don't see much of practical use of this. However, I think every biochemistry student has to read this because this is one of the earliest, easy to understand, and is an important teaching aid. So what Chou and Fasman did is that they took 15 proteins, not a very large number, with known experimental structures. From there, they defined certain rules of secondary structures, and which then they applied to unknown sequences. This method is extremely simple to implement, and it would be a good practice for a student to write down the basic computer program for the Chou-Fasman algorithm as a do-it-yourself secondary structure prediction method. So essentially the method is like this, that one first defines certain propensities for each amino acid to pick up a particular secondary structure. Say for example, if an amino acid has to, let's say, a propensity for the alpha helix. Now this is given based on your database search, how many times that amino acid occurs in a helix divided by how many times it occurs at all. So the conditional probability that the amino acid I occurs in a helix divided by the probability of existence of the amino acid. So that itself is the propensity. Okay. So once we have defined these propensities, so what we can do, we can then run through. This is an example of calculation of the propensity. Just for example, here we have 100 serine residues which are part in helices, and there might be about 10,000 residues which are in a helix, and overall serine occurs 500 times out of 20,000 amino acids. So 100 by 10,000 divided by 500 by 20,000 gives you a serine propensity of 0.4. Now, given that we have calculated these propensities for the 20 amino acids, what we do now is that when we take our test sequence, we have to assign the secondary structure. How do we do this? Okay, but before that, I must also say that in turns is a little special thing, right? Because in turns, say in a beta turn, the 4 positions are not really identical. So they have different roles to play. So one would have to calculate the propensities for each of these 4 positions. So that is also used. So let's say we have the list of these propensity values, which you have obtained from a database. Chou and Fasman used just 15 proteins, but you can use larger numbers. Now for a 3-class prediction, or 4-class prediction actually, for every amino acid, you have a propensity for the alpha helix, you have a propensity for a beta strand, you have a propensity for a turn, which could be again position dependent and all that. And coil is not really predicted. Essentially, if you predict everything else, and whatever remains will become coil. So these tables you have. Now once you have that, Chou and Fasman assumed that for every secondary structure there is a nucleation site. So for example, for a helix, he assumed that there must be a nucleation site of about 6 residues. So this you have to first identify. So what do? You take a 6 residue window and you average the helical propensity of the amino acids in the 6 residue window. And where it is higher than a certain threshold, this is considered to be the nucleation site for a helix. Now once you have found out the helix nucleation site, you can then extend it towards the N terminus and C terminus, and by again another rule which says that you take 4 contiguous residues which are just off the N terminus or the C terminus of the helix, take the average helical propensity for that, and that must be greater than 1. If it becomes less than 1 at a certain point, consider the helix to have ended at that point. Okay. So, and then again the final average of the entire thing which you have predicted thus far, that the propensity must be greater than 1. So if all these criteria are met, then you have a helix. Similarly for beta strands, and similarly for turns. So in case of beta strands, the nucleation set is thought to be just a stretch of 5 residues, 3 of which must have a propensity greater than 1. And again, extensions is done in a similar way as in case of helix. For turn, so for every tetrapeptide starting at position say I, we compute the propensity, and for coming into a turn for all the 4 residues, right? And the propensity for turn, the average propensity for turn must be greater than 1 and must be greater than the propensity of the same region becoming a helix or a strand. Okay, and then again you take the product of all of them, and if it is bigger than a certain threshold, then only the peptide is going to be called a turn. So these are the Chou-Fasman rules, which essentially just from one database you create the rules and then you apply to your sequence, and it's a simple arithmetical formula, easy to make a computer program out of, and therefore you can make a prediction. Now let's take the other one, which is due to 3 scientists, Garnier, Osguthorpe and Robson, and so the method is known as the Gore method after them. Now what, in a certain sense, it is not very different from the Chou-Fasman method. However, instead of simply calculating propensities based on occurrence and all that, they use certain information theoretic measures. The original version, this program has over time changed itself quite a few times, and the original one was called Gore 1, the current version is called Gore 4. So again, you just like the way the propensities are calculated. So you calculate a information theoretic measure, I S of R, where the probability P S of R gives you the probability of getting secondary structure S given the presence of residue R. Okay, and then one defines an information gain measure. That is, what is the information gain if I assign the particular amino acid to a particular secondary structure, right? Minus if I do not do that. So where I gain is I S of R, saying that I residue R coming in secondary structure S, minus the information where the residue R is not in the secondary structure S. So the difference between these two quantities is the so-called information gain. And then this information gain measure is summed up over a sliding window, this time of 17 residues, and the secondary structure assignment which gets the highest gain is assigned. So just you do that for helix, for turn, for strand, and whichever value is highest, you just simply assign that. So you just run through the protein like this. So if we take Chou-Fasman and the Gore method, now the advantages are very simple to implement, with rules that are fairly understandable, make sense, and it just takes a single sequence as the input and you just run it through. But the disadvantages is that at the end of the day, it's not very accurate. Now you get a Q3 accuracy of only about 50% or 60% if you are very charitable. Note that you get a 33% accuracy merely by randomly close your eyes and predict helix, strand or turn. So something with about 50 to 60% is definitely better than random, but for practical use, for biologically being useful, this much is not good enough. So we need more sophisticated methods to do something which is really becomes biologically useful. So one of these, there are very many methods for predictions from complex data, and the ones which were most popular and are still popular nowadays are the neural networks. These are computational engines that can be trained to recognize patterns from a training set which contains both true and false examples. That is, if I want to train a neural network to predict an helix, I will need a lot of different examples of helices and those which are not helices. Once trained, a neural network can be used to recognize the same patterns even in an unknown input. So this slide shows you the architecture of a neural network. It's a multi-layered thing. At one end, you have the so-called input layer, and at the other end, you have the output layer. In between, there can be one or more so-called hidden layers. Now, moment then input layer, a neuron at the input layer gets a signal, it is transmitted to every neuron which is at the higher level. So from the input layer, it goes to each and every neuron in the hidden layer, and so on and so forth, and from there to the output layer. But while this information is transmitted, there it might be reduced in intensity or increased in intensity or modulated in certain ways, and this is done by a series of weights. Now for correct prediction, what one has to do has to find out the right sequence of weights, both between the input and the hidden, and between the hidden and the output layers, and so that you predict as sequences, it gives you the correct signal to the correct neuron. And in a 3-class prediction, for example, we have 3 output neurons and gives the maximum signal to the correct prediction. So the whole problem of training a neural network is finding the correct weights in each of these links from the input to the hidden and from hidden to the output. Now, before anything, we have to have a process by which our sequence has to be coded in a certain format so that the neural network can read it. And most popularly, one uses a binary network. Now, since there are 20 amino acids, one can use a 20-character binary system to encode an amino acid. For example, for alanine, I may use 1 followed by 19 zeros. For, let's say, cysteine, I use 0, 1, and then again followed by 18 zeros. So for each amino acid, we define a specific position, which will be 1, and all the remaining will be zeros. So like that, I can define a specific code for each of the 20 amino acids. Similarly, for the secondary structures, I can define a helix as 1, 0, 0, a strand as 0, 1, 0, and a coil as 0, 0, 1. So if I am doing a 3-class prediction. So in this kind of situation, our network requires a 21-bit input for a window size of n, where 1 bit is required per residue to indicate that the window overlaps with the end of the chain. The advantage of this sparse encoding scheme is that it does not pay attention to the ordering of amino acids. Okay, so it does not matter which way the amino acids are ordered, you can use this encoding. But the main disadvantage is that it needs a lot of input. So for every amino acid, it uses a 21-bit input. So 21 times increase in the sequence length will happen. So this is an example network. For every amino acid you take, which is on the extreme left of the figure, and then you create the corresponding bit pattern, and this is then transmitted to the neural network and processed further. So again, as you can see, various encodings of amino acids are given. So each amino acid is defined a particular position in the bit vector, and that one is set to 1 while the others are set to 0. Now the question of these weights. Now the input value at each of these layers are multiplied by the weights, and that either can raise up the signal or it may reduce the signal, and then it is transmitted to the next level. And the weights are adjusted after the output is computed based on how close the output is to the right answer. Now what I do during training, I give inputs with known sequences for which I know the secondary structure, and I measure what is the output. Now let's say I have given the sequence for a helix and the output says turn. So I know that something is wrong, I do not get a correct one. So what I do, I readjust the weights so that this particular set of weights which I have given to a wrong prediction does not happen. If on the other hand I train, during the training I input an helix and I get a helix, then I know this is working. So I will try to strengthen this set of the weight. So this training session goes on with iteratively taking different positive and negative examples and obtaining a set of weights which always correctly predicts both the positives and as well as the negative. So where the number of mistakes are minimized. Now given this set of weights, which once I have had it from a trained network, then I can just take an unknown sequence, run with the same set of weights and try and see what is my prediction. Now to understand this in more detail, let's look at just one unit of a neural network, which is called a perceptron or a single neuron. So here again, so this is the neuron and it is getting signals. So in this case I show 3 from all the neurons from the previous layer, but each of them is weighted differently. Okay, so the first one is with weighted W1, second one is weighted W2, third one is whatever, the nth one is weighted Wn. Now given these signals, these are added up or manipulated in a certain way, and so as you can see here, so there is a sum over an x times W, and there might be even more manipulations out there, and the output is a single binary 1 or 0. So now let's say I take 3 amino acids and I have some random weights to start with, and it outputs 0. Now if these 3 amino acids are helices, and which I know that they are part of helix, and I expect a 1 for a helix, then my output is wrong. So what I will do, I will adjust the weights so that I get output 1 instead of output 0. So this way I will correct for the weights for every perceptron as it is going on and in the entire network, and once the whole thing is done, I expect to get a correct prediction most of the time. So mathematically what do? I find out that set of weights which minimizes a certain error function, and this minimizations can be done by gradient descent methods, and which is done by using calculus to find out the n-dimensional gradient of the error function, and then there is an update rule by which we can go on iteratively improving the weights until we get that weight which minimizes or reduces the error function as much as possible. So the advantage of neural networks is that it can handle information from neighboring residues, okay, that depends upon how wide the network you make, in a very generalized and non-linear way. So whatever kind of interactions they are, it's kind of taken in. Its disadvantage, on the other hand, that it's a black box. Now, I would like that if I have a trained neural network which is predicting, let's say, helices perfectly every time, I would like to know that what rules it sees actually. That would help my science. Unfortunately, dissecting out a neural network and finding out that what rules it has actually seen is a very difficult task. So it's used more like a black box. So even though if it works correctly, we do not know why it works correctly. And similarly, when it doesn't work correctly, we are not very sure why it didn't work correctly. So that is a little bit of a problem. So a few things to be noted. That the network should be trained in a way that it can generalize the input pattern and look for them in unknown sequences. Okay. Sometimes unimportant patterns are also learned. Sometimes we call them memorizations or overtraining. In such a case, the network performs very well for test sequences and performs very badly for unknown sequences. So there are techniques to prevent overtraining, commonly cross-validation, even multi-layered cross-validations, etc. are used as a guard against overtraining. So whenever one is training a neural network, one has to be very, very careful so as not to overtrain the system. This can be done by minimizing the size of the network, so that minimizes the number of parameters which have to be adjusted, or increasing the number of and the diversity of test sequences by which this training sequences by which this training has to be done. So there are various approaches, and using robust cross-validation methods to make sure to stop the training moment it is getting towards overtraining. So now, one of the best neural network based methods is the so-called PhD method due to Burkhard Rost and Chris Sander. So what it does, it uses neural networks and combines them with sequence profiles. We had earlier discussed sequence profiles in the talk on pattern recognition on sequences. So what it does, one sees that there is about 6 to 8% increase over just use in performance just using neural networks. That is, if we use the neural network and the sequence profiles, then you get about 6 to 8% better. And also the networks are, they are divided. So there are different networks which are predicting, and then there is a consensus, a jury of predictors is used. So maybe 5 independent prediction systems are used. 3 of them says it's a helix, then you pick it up. Well, 2 of them might say other ways also. So given the uncertainties in individual neural network performances, so using multiple such networks and then using a jury to or a consensus to come to the correct answer always gives you better results. So this is an example that you have the data, and then it goes into this complex neural networks, and also sequence profiles are used, increasing the information that can be gleaned. This improves prediction. So again, it gives you a 3-class predictions. Okay, so using from sequence to structure, it can predict alpha helices, beta sheets, and coils. And each of these, this prediction system is divided into a set of different neural networks, and for each residue, you take a certain window of that, and then you put it to a neural network, get a prediction. These predictions are then fed into another neural network, which with a different window size, and finally the final output is what is taken. The critical thing out here is that the whole process is repeated with independently trained neural networks, and the results are averaged or consensus taken so as to minimize idiosyncratic errors from one or the other network. Now there is another method called the Psi-Pred due to Jones. This uses iterative sequence alignment technique like Psi-Blast as an input, and this is then put into a neural network. So it starts with the alignment based kind of technique and is done best with Psi-Blast, and that result is then fed into a neural network. So it takes both of the sequence based and the network based predictions and to make correct predictions of secondary structures. Again, the Psi-Blast profiles which are obtained are encoded, made into a binary scale with values between 0 and 1, And once that is done, again a window-based approach is taken with 15 to 20 residues per window, and these are then fed into a neural network. Again, just like the PhD kind of thing, you have a lot of different neural networks which are fed into it. The output of one goes to another layer of neural networks, and you get a final 3-class predictions.